It's Misonto Sane, student from the Wonkong University, South Korea. Today I'm going to explain about the elevated plasmids, which is used to measure the level of the anxiety in the rodents. Today I will explain about the detailed procedure about the elevated plasmids in this video. Anxiety is the psychological and emotional feelings characterized by the uncontrolled feeling of worries, restlessness, poor concentration, insomnia, dizziness, faintness, and increase in the blood pressure and heart rate. However, despite its unpleasantness effect, anxiety in the small dose is clearly necessary for the performing everyday activities. For example, anxiety before the exam encourages us for the study. Also, anxiety before the public speaking forces us to practice. And its principle. The elevated plasmid test is one of the most widely used tests for measuring anxiety-like behavior. The test is based on the natural aversion of the mice for the open and elevated area. It means that the mice don't prefer to go to the open place and also in the height. The elevated plasmid is widely used behavior assay for the rodents, which help to record the duration and frequency of entry into the open and closed arms. The test is based on the approach avoidance conflict, meaning animals have the tendency to struggle for the novel environments and unconditional fear of high and open space. Likewise, if the animals decrease the number of the entries into open arm and the time spent in the open arm, then they are said to be the anxiogenic animals. As I have already mentioned that the elevated plasmids have 4 arms, 2 of them are open and 2 of them are closed. It consists of the open space in the middle. Animals are kept in the central place so that they can face all 4 arms. The animal are allowed to explore arms for the 5 minutes and 1 minute is given for the adaptation time, which is recorded in the video. Similarly, the total number of the entries and total time is spent in the closed and open arm are recorded from the video. This is the experiment I did in my lab. The PanLab smart software detects the mice mobility by tracing the mice mobility in open and closed arms, which also record the total time and the total number of the entries made in each arms. Finally, PanLab software converts this data in the Excel format. As I have already mentioned that the anxiogenic mice decrease the total time in open arms and open arm entries, whereas increase total time in closed arm and closed arm entries. Here in my results, the test compounds show significant increase in the total time in the open arm and open arm entries, which means that the test compound have the anti-anxiety activities. After getting the positive result from elevated plasmids, we further decided to analyze the biochemical and molecular signaling pathway by using the Western blot, protein analysis, and HPLC neurotransmitter analysis. We took the brain of the rats and the mice and then further analyzed in the biochemical and molecular signaling. Here in the Western blot, there is significant increase in the tyrosine hydroxylase by the test compound. As we know, the tyrosine hydroxylase is the rate limiting enzyme for the synthesis of the dopamine from the amino acid tyrosine. Also, Western blot results show that the GAT67 is significantly increased in the test compound. GAT67 is the glutamate decarboxylase, is an enzyme that catalyzes the glutamate to GABA. As we know, the GABA is considered as the inhibitor neurotransmitter because it blocks or inhibits the certain brain signal and decreases the activities in the nervous system. It is simply considered as a calming neurotransmitter which creates calming effect to our brain. Then finally, after Western blot, we analyze the mice brain by using HPLC analysis. Here the HPLC analysis shows that both the dopamine and the serotonin level will significantly increase in the test group as compared to the control groups. These neurotransmitters are important for the human beings for well-being, happiness, and to produce the calming effect. The purpose of the elevated plasmase is given as pre-screening of the newly developed pharmacological agents for the treatment of the anxiety-related disorder, to investigate the anxiolytic and anxiogenic effect of the pharmacological agents, drugs, and hormones, can be used as a behavior assay to study the brain sites such as limbic region, hippocampus, nucleus accumbens, stratum, and also the mechanism that how they regulate and modulate the neurotransmitter and hormone level in the brain. For example, GABA, glutamate, dopamine, serotonin, corticosteroids, etc. Here I try to show the clinical application of the elevated plasmase to the human beings. Here the scientists try to create the virtual and the real world elements which is shown in the video.